Hi, everyone. My name is Sonam, and I work at UCL, and I'm also part of the Patient and Public Engagement BSGCT subcommittee. And today we have Professor Nicola Brunetti-Pierri. Sorry, the name is probably not quite correct pronunciation, but we're really delighted to host you today and hear about the liver-directed AAV gene therapy. But before that, I just want to mention about how to submit questions. So again, we have the live chat window where you're more than welcome to put your questions. If not, feel free to email raj@r.card@ucl.ac.uk as well. And yep, and if you could put your name and affiliation in the question as well, that'd be very helpful for us. So without any further ado, I want to introduce Nicola. So we've worked together, at least with Julian, and I've had the opportunity to work with Nicola through Julian as well, which has been a great experience. And Nicola's preclinical and clinical research focuses on the elucidation of the pathogenic mechanisms and development of new gene therapies for genetic diseases. And he has a long-standing interest in liver-directed gene therapy. He mentioned he's been doing this for almost 20 years. And he has been the principal investigator of phase 1/2 liver-directed gene therapy clinical trials, which we'll hear about today. And he's also professor of pediatrics at Federico University of Naples, and also PI at the Italian Institute of Genetics and Medicine in Italy. So yes, Nicola, I'll let you take the floor. Thank you.
Thank you. Thank you, Sonam. And thank you for Raj for the nice introduction. As mentioned, the focus of my work is on inherited metabolic diseases. This is a large group of different disorders that can be due to defects in one of the many metabolic pathways that are operating in our body. This understanding inherited metabolic diseases that are Mendelian, these single gene disorders, have been instrumental to understand the metabolism in general. And there is also the concept of a complex disease that can be due to impairment of multiple metabolic pathways, and therefore understanding Mendelian disorders can be also instrumental to understand complex diseases. I mean, the inherited metabolic diseases were really discovered by Garrod, shown in this picture, at the beginning of the 19th century. Back then, there were only few disorders, but over the years and the decades, the number of diseases that have been discovered has been increasing exponentially. And this is now a group of diseases that includes over 1,000 of different diseases. For most of these diseases, the treatments are completely absent, or if they are present, they are often unsatisfactory. And therefore, we need to develop treatment for many of these diseases or improve treatments for those that have available treatments now. And the liver, and particularly the main cell of the liver, that is the hepatocyte, plays a very important role in metabolism. It has been estimated that over 5,000 metabolic pathways are operated by hepatocytes. And this is the reason why hepatocytes is also a very important target for gene therapy. I want also to mention that this group of diseases is historically important because actually the very first example of some of the very first diseases in which gene therapy has been tested and developed have been inherited metabolic diseases. So liver gene therapy, therefore, is an important area of research because the liver is affected, as I mentioned, in many inherited metabolic disorders, but also because the liver is relatively accessible to vectors delivering genes that are infused by simple and safe intravenous infusions. Because of the liver microarchitecture, the blood is in direct contact with the hepatocytes, and therefore liver is very well accessible to blood-borne gene therapy vectors. Another advantage of the liver is that the hepatocytes can be also used for production and systemic delivery of proteins that have therapeutic functions even outside of the liver. Many inherited metabolic disorders are treated by liver transplantation. There is a defect in an enzyme that is operating in the liver cells, and the liver transplantation is a very simple but brutal approach in which thousands of genes are replaced with a whole organ just to correct a single gene defect. The gene therapy, in contrast, is a more elegant and sophisticated way to correct a disease-causing gene defect by simply delivering the gene that is not properly functioning. And therefore, for many diseases, gene therapy can be viewed as an alternative to the invasiveness of liver transplantation. One of the vectors that emerged as the most attractive for liver-directed gene therapy has been the adeno-associated viral vectors, AAV. The advantages is that these vectors are deriving from viruses that are non-pathogenic, so are not causing any diseases or illness in humans. They can deliver genes to different cell types, including the liver cells. There are multiple serotypes that are available, and they can also drive long-term expression of the therapeutic gene after a single administration. Most importantly, they've been shown to be safe in humans. Disadvantages of the AAV vectors is their cargo capacity. Their genome is relatively short, and they cannot deliver large genes. This is, for example, a problem for diseases such as Duchenne muscular dystrophy, CFTR, the gene responsible for cystic fibrosis, and Wilson disease, that are due to genes that are too large to be included into an AAV. But it's not really an issue for the great majority of inherited metabolic diseases that are due to defects in genes that are small and can be easily accommodated into AAV vectors. That you see here in a 3D pictures how that vector will look like, in particularly the capsid, how it will look like. Within the capsid, there is the genetic material that will be delivered to cells. So once the vector is infused systemically, it will make a long journey, going first into the lung capillary bed, then it will go to the heart, and from the heart will be spread systemically. But even though the capillary of the livers are not the very first organ that the particles will encounter, the vector goes very efficiently to the liver cells and transduced very efficiently to the liver cells, because the liver has a almost unique microarchitecture in which the capillaries that are shown here and made by the endothelial cells, of course, they have fenestration. So you can see here there are holes between the endothelial cells. Those are called, again, fenestration. And typically in other organs, there are no holes, there are no fenestrations, and there is even a basal membrane underneath the endothelial cells. So there are, in other organs, physical barriers that are preventing vector particles to come in contact with the cells of that particular tissue. But this is not the case in the liver. You can see that here some small vector particles are going through the fenestrations, and then they can deliver their genes to multiple cells. So you now saw some of the cells that are turning green, and then means that they've been transduced by the vector. For most of the genetic conditions, we don't need to transduce 100% of the hepatocytes. And actually, the percentage of hepatocytes to be transduced in different disorders varies according to the specific features of the disease. For example, diseases such as hemophilia, a bleeding disorder, requires only a very small percentage of hepatocytes to be transfused to convert a severe phenotype into a mild phenotype. And this is why there is plenty of studies about liver-directed gene therapy in hemophilia, and there are recently also 2 approved products based on AAV gene therapy for hemophilia A and B. Another disorder in which a small percentage of hepatocytes is needed for correction of the phenotype is called the Crigler-Najjar syndrome, and I will be talking about this disorder later in the presentation. Then there is the group of the lysosomal storage disorder, such as the MPS or mucopolysaccharidosis, that requires a relatively small percentage of enzyme for obtaining clinical benefit. But there are other diseases, such as the urea cycle disorders, disorders of the lipoproteins that may require much higher percentage. Of course, the higher percentage of hepatocytes needed for achieving a clinical benefit, the higher is the dose of vector that it's needed. And diseases that requires higher percentage are for this reason more challenging as compared to diseases that require less hepatocyte transduction. And I will now focus on one of the mucopolysaccharidosis. Is a group of diseases within the larger group of lysosomal storage disorder, and it's called MPS, mucopolysaccharidosis type 6, or also known as Marotola-May syndrome. So this is one of the several types of MPS that is due to a specific defect in the enzyme aryl sulfatase B, ARSB, and as a result of deficiency of this enzyme, there is an accumulation of the substrate of the enzyme that are called glycosaminoglycans or GAGs. This is a multi-system disorder. Basically, every tissue and organ is affected, but the brain is spared. This is important because lysosomal storage diseases that require that have also brain involvement are more difficult to be treated. And this disorder is currently treated with weekly infusions of the enzyme, the aryl sulfatase B. And a few years ago at my institute, TGEM in Naples, studies were performed to prove that gene therapy was effective in animal models of this disorder. I'm only showing one piece of this data in a large animal model of MPS 6. It's a natural model of MPS 6 that is showing multi-year expression following a single AAV infusion. So you see here 3 cats showing normal levels of RSP for many years after a single IV infusion. Very different from the affected range. And of course, this speaks to the potential of the gene therapy that may provide a source of enzyme that will be sustained over time. This is this experiment was performed using an AAV that was delivering an expression cassette made by the TBG promoter, that is a liver-specific promoter, driving the expression of the RSP. And in this experiment, what is happening is that the liver cells are transduced, and the hepatocytes in particular are making the aryl sulfatase B, are secreting the aryl sulfatase B. That is what we measure here in the blood of the animals. And this enzyme can then correct all the peripheral tissues that are affected by the disease. So in this experiment, really, the liver is a factory organ making the enzyme that is needed for a correction of the peripheral tissue. And so this is the principle in which we have a genetically modified cells, that is in our case, the liver cells that is making the enzyme. Then the enzyme can be secreted, and because lysosomal enzymes are naturally uptaken by cells, this enzyme made by the genetically modified cells can cross-correct other tissues. So this is a feature that is unique of lysosomal storage disorder, and we take advantage of this natural capacity of cross-correction of uptake of enzyme by peripheral tissue for therapeutic purposes. So based on the encouraging preliminary data, So we decided several years ago to move into the clinic with the same approach. And we developed a trial that is a phase 1/2 open label trial for intravenous, evaluating efficacy and safety of intravenous injections of the AAV8. So a specific serotype of AAV8 that transduces deliver quite well and is driving the expression of the aryl B. The plan was to inject 9 subjects with the age above 4. And because this is a phase 1/2 study, we had to look at safety as a first goal. But we also look at secondary objective as a secondary objective at efficacy at the clinical and biochemical level. As with any trial, we had the inclusion and exclusion criteria. I just want to draw your attention to the fact that we enrolled only patients that were under ERT, because that's the standard of treatment, available in all patients diagnosed with this disorder. But the ERT that was discontinued 7 to 14 weeks before the infusion of the gene therapy vector. And also, we excluded patients that had antibodies against the AAV8. Because the AAV can cause, we are exposed to AAV in nature. That is because these are viruses that can infect us. Although we don't develop any symptoms following an infection with the AAV, the natural infection with the AAV, however, will result in the formation of antibodies against the virus, and those antibodies will prevent the infection of the AAV towards the hepatocytes, and so preventing therefore the delivery of the gene, the therapeutic gene, to hepatocytes. Therefore, we excluded patients with this type of antibodies. So these are the patients that we enrolled. And those are 9 patients. This is a phase 1 study, so it's a patient-by-patient study. The follow-up is different for all the patients because we could enroll the second and third and fourth patient and so on only after data about safety from the previous patient enrolled were obtained. And as you can see, we have patients that received the low dose, and those were 3 patients. 2 patients with intermediate dose, and then we had 4 patients with the high dose. And I will mostly talk about the 4 patients with the high dose, that is a 6 times 10 to the 12 genome copy per kilo in this presentation, because those are the ones in which we see the best outcomes. So this is and we have not seen any problems with safety, and therefore the goal of safety was met. And then besides safety, we look, as I mentioned, also at the biochemical measurements of efficacy. And so this is a very busy slide, but I will try to walk you through the slide. So if you focused on the left part of the slide, okay, the left part of the slide shows the levels of the enzyme in the blood. As you can see, all patients are starting with almost zero activity of the enzyme. And following the injection performed at time 0, we can see an increase in enzyme. This is good news, of course, because it shows that the gene transfer indeed occurred in all patients, with the exception of 1 patient that showed no detectable level of expression. In black, you can see the subjects that received the low and intermediate dose, and the levels, as you can see, are much lower as compared to the other patients at the high dose that had higher level of expression. They did not reach the healthy reference range, but they were at about 50% of the normal. Then on the right part of the graph of the slide, you see the urinary GAGs. So the urinary GAGs at the baseline are affected by the fact that these patients have been under ERT up to the gene therapy. So, but the ERT was discontinued, and in fact, in the patients that received the low and intermediate doses, so the ones that are shown in black, we can see a quite rapid increase in urinary GAG, suggesting that the amount of enzyme that was produced at a low and intermediate dose was not sufficient to maintain the GAG within the normal level, or at low levels in general. Instead, the patients that were receiving the higher dose, that had the higher levels of the enzyme, had a better control of the urinary GAG. There was some modest increase in urinary GAG, but they maintained relatively at a relatively modest level, below the threshold of urinary GAG that is observed in untreated subjects, that is shown by the dotted line. Only one patient, that is shown in green, was recommended to restart the ERT because of that, of some data points that were of urinary GAGs that were above the threshold of urinary GAG in untreated patients. And this was a recommendation given even if the patient was doing clinically perfectly fine, but for the sake of the safety, we recommended to restart the ERT. And as you can see, both the level of enzyme and the urinary GAG have been maintained stable for several years now, following the administration of the vector. And this was accompanied also by stability in the endurance. So one of the features in this disorder is that the 6-minute walking test and 3-minute state climbing test that are testing endurance are progressing. So patients will get worse over time with impairment over both tests. And these were also tests that were used in the past to get approval for the ERT. And interestingly, as you can see here, the endurance have been maintained, sustained, and relatively constant over the years, despite these patients are not on the ERT anymore for many years. So we concluded that the gene therapy for MPS 6 with the AAV was safe and well tolerated at all doses, meeting the safety goal of the trial. With the low and intermediate doses, the amount of enzyme was not sufficient to maintain control of the urinary GAGs. But at the higher dose of 6 × 10^12, the amount of enzyme was higher and the urinary GAG increased, but in a modest manner in 3 out of 4 patients. And in the high-dose cohort, we see clinical stability in terms of endurance that I showed, but also with other by monitoring other organs that are typically affected by the disease, such as the liver and spleen, that typically are enlarged in the disorder, and cardiac function. And over these 4 years after gene therapy, patients have been able to interrupt the weekly and inconvenient and costly infusions of the enzyme. I will now move to the other disorder in which we have been involved with a clinical trial, that is called Crigler-Najjar syndrome. So this is a rare disorder of bilirubin metabolism. So it's a disorder due to the deficiency of the enzyme that is responsible for conjugation of bilirubin with the glucuronic acid. This conjugation is essential for excretion of the bilirubin. If this enzyme reaction does not occur, there's an accumulation of unconjugated bilirubin that can cause brain damage that is very severe, maybe even life-threatening. In the old textbook, this is described, the brain damage induced by bilirubin is described as kernicterus, because if the autopsy is made on patients that died because of this disorder, there is a yellowish coloration of the brain. And the jaundice is also, of course, because of the increase in conjugated bilirubin, the main feature of this disorder. The patients with this disorder are currently treated with the phenobarbital only in cases that are responsive. So typically patients that that have milder mutations that results in residual activity that can be further increased by inducing hepatic enzyme with phenobarbital, or patients have to spend several hours per day, up to 12 hours per day, under the phototherapy or the UV lights, because the UV lights allows the conjugation of the bilirubin into an isomer of bilirubin that can be excreted in the urine. Because of the inconvenience of spending so many hours under the bilirubin light, many patients decide to have the liver transplantation. And the gene therapy can be viewed in this case, of course, as an alternative to the liver transplantation. And after preclinical studies showing efficacy of a gene therapy by AAV in the animal models of Crigler jar, a few years ago, within an international collaboration with other centers coordinated by Genethon, we started a clinical trial that is quite similar to the trial I previously described for the lysosomal storage disorder, because it's based on the AAV8. Of course, in this case, it's delivering a different therapeutic gene. Here, the goal is to enroll 17 subjects with the age above 10, with the 2 doses, 2 times 10 to the 12 or 5 times 10 to the 12 vector genome per kilo. Again, this is a phase 1/2 study, therefore is investigating safety as a primary objective and efficacy as a secondary objective. And measurements of efficacy are based on very simple determinations of bilirubin in serum and the hours under phototherapy, because the goal is to allow these patients to interrupt the phototherapy. So these are 6 patients enrolled in this trial. Just by chance, they occur to be all females. This is their genotypes. Of course, they have biallelic mutations in the gene encoding for the UGT enzyme. And as you can see, they need at their baseline several hours of phototherapy to maintain the bilirubin below a threshold that may be dangerous for brain damage. And they've been enrolled to receive either the low dose, the 2 initial patients, or the next 4 patients, the high dose. And here are the results of the low and the high dose as far as the bilirubin. So let's start from the serum bilirubin level. You see here, patient 1 had a drop, a very interesting and encouraging drop in bilirubin level, but this drop was only maintained for weeks, but then was completely lost by about 12, 14 weeks post-injection. So there was only a transient effect. On patient 2, the reduction of bilirubin was mild, but was not maintained. And patients also returned to baseline bilirubin after several weeks. And the interruption of phototherapy was not even possible, except for a small period of time in patient 2 in the low-dose cohort. In contrast, patients that received the higher dose had a significant reduction in the bilirubin level, as you can see in all 3 cases, that was maintained for several weeks post-injection, and they allowed the suspension of the interruption of phototherapy at about 18 weeks post-injection. So very encouraging, and actually this had, of course, as you can imagine, a significant impact on the quality of life of patients that could stop the night, all those many hours that they used to spend under the phototherapy. And I want also to show you the results on the patient that I infused with the gene therapy vector, because I think this was one of the one memorable, I think one of my best day in my career as a physician, when I had the opportunity to treat this patient. So as you can see, the patient has severe jaundice. You can see the sclerae that are quite yellowish before the gene therapy. When the patient came for the first visit post-gene therapy, her eyes were looking like this. So the sclera was completely white. And by not even doing any blood test, we could tell that the gene therapy was working. And again, the day she came post gene therapy was a day that I will never forget, because we were all happy and celebrating this fantastic result. Of course, when the lab results came back, we had they documented a very rapid reduction in the bilirubin levels. So as a conclusion of this second clinical trial, the patients received AV8 with a good safety profile. And at the high dose, we did see a signal of efficacy. We didn't see, of course, the signal of efficacy at the low dose. And the patients that received the high dose were able to interrupt the phototherapy. This is all looking very encouraging, but of course there are still open issues with AAV gene therapy. One of the issues is about the neutralizing antibodies, the pre-existing neutralizing antibodies. I alluded to this problem when I mentioned the exclusion criteria. So there are individuals that have, simply because they came in contact during their life with AAV, they have these antibodies that are preventing them to participate to gene therapy. There are multiple ways, multiple strategies that are under investigation trying to overcome this issue. Another issue that I didn't have really the time to talk about is about the CTL immune response. Basically, the immune system recognized hepatocytes that have been transduced by the AAV vectors and try to clear them. And to avoid this immune-mediated clearance that has an important impact on the efficacy, there are different protocols for either prophylaxis or intervention with the steroids. Although we don't really know which one is the best approach and which is the best immunosuppressant to be used. There are diseases in which early intervention is needed, but if the AAV injections is performed too early, there is a chance that the vector will be lost over cell division, because this is mostly an episomal vector that is not maintained with the cell growth. This is a similar issue with the liver growth. There is loss of expression. Therefore, if there is any replication of hepatocytes, for example, as a result of an insult to the liver, also the expression from the vector can be lost. And last, there is a concern about malignancies, particularly in the liver, that have not been shown so far and have not been documented so far in humans, but they have been seen in animal models under specific circumstances. For example, when the injections are performed in mice in the newborn period at extremely high doses. There has been clonal expansion seen in dogs, but no hepatocellular carcinoma in dogs so far. But there is some concern that there could be an increased risk of malignancies as a result of AAV vectors. So in summary, the clinical studies are showing promising results. I think that is pretty obvious, with also products that have been approved, like in the case of hemophilia. There are still several issues to be addressed, like the immune response, long-term safety, having a higher efficiency of liver gene transfer, particularly for some diseases that require higher percentage of liver transduction. There is also a challenge in gene transfer to diseases that have altered microarchitecture, for example, fibrotic livers. And there is also, finally, even if the product is approved, there are issues with commercializations that is due to the very high prices that this product may have. But there are multiple strategies that are being investigated to overcome these obstacles. With that, I will finish by acknowledging the people that participated at this trial. So our clinical team and people at the Teleton Foundation that helped us with the development of the clinical trial protocols and with conducting the clinical trial. All the other physicians and colleagues that participated to the trial and the funding, and of course for the MPS 6 and as well as the Najar study. And of course, we also thank the patients for participating to the study. Sometimes patients were coming from different part of the world for the infusions, so they make, you know, there were patients that were brave enough to participate to the first in human study, and we are grateful to them as well. And thank you all for your attention.
Thank you, Nicola, for that amazing talk. Sorry, I got cut off
Yeah.
my side. Thank you, Solon, for stepping in.
That's okay.
So those are two very amazing studies for clinical trials that you've talked about. I think my question to you would be, because gene therapy is starting to become quite of a common form of treatment for a lot of these kind of very severe genetic disorders, and we're kind of still learning as these studies are coming through with your two studies you've discussed. Do you feel like there'd be need for longer follow-up? Because the last time storage study you discussed, there was only a 4-year follow-up period. Would these patients be followed up for the rest of their lives to see how they are dealing with the gene therapy, or to assess whether or not they need to go back onto any drug therapy if that's the case?
Yeah, definitely we will need a longer follow-up. And so there are recommendations of follow-up with a minimum of 5, even 5 plus 3 years post-gene therapy. I think that should be even longer. And not because we need to know the absolute duration of this of the expression coming from the liver that have received the vector. It may well be that the expression at some point would be lost. Hopefully this will happen after many decades, but it could be that it will be lost at some point, because the hepatocytes have very low turnover, but they do have some turnover. So, and if hepatocytes are dividing, the vector genome would be eventually lost. So we definitely need to look at the longer follow-up, both for the efficacy as well as for the safety.
Yeah, great.
And I had a question. You mentioned about because there could be transgene loss as the liver grows, so targeting them too early on might not be as effective. Would you recommend them to just be on some form of management kind of diet or something before they actually receive the gene therapy? Or could they be in other form of therapies before receiving AAV when they are more adult stage?
Yeah, so thank you, Sonam, for that question. So I think it varies a lot from disease to disease. Unfortunately, there are some diseases in which you need very early intervention, and currently available interventions are very much unsatisfactory. And those are specific cases in which even early gene therapy may be considered, I think. Whenever possible, well, at least with the vectors we have today, the ideal situation is to manage them with the standard treatment available until the liver growth is completed, and then go with the gene therapy. So whenever that's possible.
Are you having more patients recruited for these? Are you having more patients being recruited for these trials? I'm curious.
Yeah, there have been more patients that recruited for the Krieger-Najjar trial.
Okay.
The MPS trial is now ended, but we are working for the phase 3 trial. So well, the next phase. Of course, the phase 3 is much more demanding and financially even more challenging than phase 1 and 2. Phase 1 and 2 is already very, it's extremely demanding, but phase 3 is a totally different story, because there we need to prove the efficacy with the large number of patients that will be enrolled to receive either ERT or a gene therapy in a randomized fashion.
Okay. So in the UK, we have I believe, about 4 treatments approved to be used as gene therapy treatments to be available to patients in the UK. And slowly, I think as time goes on, the gene therapy field will start to grow and more treatments would slightly start to become more available and accessible for patients in the UK. So I was wondering, how is the situation in Italy? Is it very similar to us? Do you have any treatments that are readily available, like AAV or gene therapy treatments available for patients in Italy? Is it a very similar system for you there?
Yeah, so I guess I think we do have a few more products rather than the 4 you mentioned. So there are the hemophilia, the 2 products for hemophilia A and B, the spinal muscular atrophy, the Luxturna for the eye.
Yeah.
Then there is the recently approved gene therapy for Duchenne.
Yeah.
And then the one for AADC. For the
Right. Okay.
very rare
Amazing.
metabolic disorder. So those are so I get the path for approval. I think is pretty much similar. In our case, it goes to the EMA.
Yeah.
And then once it gets approved by the EMA, is the national authority, drug authority in Italy will then follow that the recommendation of EMA, and most of the time also it will be consistent with the decision of EMA.
Okay, great.
And I had one more question, just again out of curiosity. But the clinical, you mentioned about the process itself for phase 1 to trials is it's a lot of work. But how was the process itself like recruiting patients? Because I guess you're already limited by the fact that it's a rare disease, and then you have further limitations with the AAV antibodies against AAV serotypes. So how difficult was it to get to this stage of like patient recruitment to like phase 1, phase 2 trials?
Yeah, that's an interesting point, because we had a bit, and I was not expecting this, but we had different challenges in the 2 disorders that I mentioned. For the MPS 6, enrollment went very well and very fast. Probably because there is a large patients community that is very well connected, and we had requests for coming to be enrolled coming from different parts of the world. Actually, patients coming from Canada, Argentina, US, Europe, of course, Turkey. So we had, and that was not difficult. So enrollment was not difficult. We of course we had to exclude about 30% of patients because of the neutralizing antibodies. But the number of patients was not really a limitation. This was a bit different with Crigler-Najjar. Instead, that we had more resistant, was more difficult. Largely because it's Crigler-Najjar is an ultra rare disorder, is more rare than MPS 6, and also, of course, the option of liver transplant also take away patients from the gene therapy. Of course, if patients have been transplanted, of course, they will not be eligible for the gene therapy anymore.
Great. I believe that's all the questions we had, wasn't it, Sonam?
Yeah, I think that was it.
Yeah, thank you very much, Nick. It's been a very informative talk, and it's good to understand how these new therapies are coming through, and also just across Europe, how it's working. You know, these treatments hopefully be available for across the world. Like you said, you're entering into a phase 3 clinical trial, and it gets more complicated. And it takes a bit more time as well to recruit more patients. So we're really looking forward to this space and looking forward to how we can start to treat more of these rare metabolic disorders. So thank you very much for giving a talk today in our series.
You're welcome.
Thank you.
My pleasure.
Thank you.
Thank you.
Thank you for the audience for joining as well. So we'll be having another talk in September, and we'll send out those details shortly. So on that note, thank you guys for listening in, and see you guys very